This is first video of in the world, whole world, I claim, which is documenting the rhizomorphs radiating from Macrolepiota procera, a very well-known, universally well-studied, almost cosmopolitan macrofungi of the Agaricaceae in the order Agaricales. Genus Macrolepiota procera, it is known as parasol mushroom, non-toxic, medicinal, very powerful source of enzymes. That we see these rhizomorphs colonizing mixed leaf litter very clearly here. These are the rhizomorphs which have colonized the leaf litter. Many times if the leaf litter is brought to the lab and examined, it won't produce a fruit body like this, and we won't even know because it is difficult to identify to which fungal species or macrofungal species the rhizomorphs belong, because they are very powerful survival organs of the Basidiomycetes, which are extensively studied by mycologists in Europe and other countries. But unfortunately, to get the documentation specific to means now here is the vegetative state or the survival state. This is the survival state. This is a very long survival state, because if it is left like this for a longer time, it will become blacker and blacker, and then it will remain there in a dormant state till moisture revives it, and then we get this. Because this is the way to colonize the Basidiomycetes, colonize the litter like this and produce this rope-like structure, which are survival structures, and they transport the nutrients and water and everything, and then they go on like cobweb invading the surrounding the litter close to the where the you can see the fruit body, that is Macrolepiota procera. So this is a very interesting example. This is also a very interesting way of looking at. This is not documented much. That what we see here is the mycelial mat which has enveloped the basal part of the stipe. Now I have not encountered this at many places during 35 years of my research work in field mycology. What it means is perhaps that because of the unfavorable condition, perhaps in the process of autolysis, perhaps the mycelia is recycling its own the tissue. I don't know, because I'll have to every day check up with this mushroom till they completely decompose. So then I'll be able to report back on exactly the sequence of events which are involved in natural autolysis or decay of this specimen. Here I'll be focusing, unless the animals at night destroy this or some human interference in this. Generally there is no human interference in this area because it is very difficult to step in. But I will be able to properly build a sequence of this decomposition as a function of time in the coming